To live is to risk it all. Otherwise, you're just an inert chunk of some randomly assembled molecule drifting wherever the universe blows you. Good day! Good night! To both the one and only Sir Patrick, the Department of Biology. Welcome to Lecture 7, Introduction to Molecular Systematics. So I'd like to start my lecture for today with some critical issues that the field of systematics is facing right now, both in the local Philippine context and in the larger international context for the field. So the first one that I'd like to mention here is the fact that most taxonomies today are growing old. So we can't deny the fact that these old taxonomies that are now retiring are leaving this what we call orphan taxa behind, with no one else to study them. So it kind of feels like a dead end for that taxa if there are fewer and fewer students that are entering the field. So that's the problem right now. There are fewer and fewer young people that are joining in the fight to try and identify and describe all of those new species that are out there in the wild. So we have fewer and fewer manpower right now. And in line with that, I'd like you guys, and maybe to inspire you young people to join in the cause, I'd like you to meet Mr. Leonard Ko, or Sir Leonard, no? Sir Leonard as he was fondly called. So Sir Leonard was a brilliant man who was the foremost expert in Philippine botany. He advocated for the protection and restoration of Philippine rainforests. In fact, I always watch all of his documentaries in YouTube where he describes species. He even, one of his techniques to identify species is to grab the leaves and crush them and try to smell if there are some scent in that leaves, or maybe break some stems to see if there are saps that are oozing, so he can deduce what species or what genera or what family is that tree or that plant is. So he is really a brilliant man back in the days. In fact, in his last day, he and his team were looking for some tree species that the future generation of Filipinos can use to rebuild our dying forest. On that day, November 15, 2010, Sir Leonard and his companions were sadly killed mercilessly by the Philippine military in the forests of Tongonan, Leyte. The military mistook Sir Leonard and his guides as NPA rebels, despite their plea of innocence. Until now, the families of Sir Leonard and his companions and his guides are still calling for justice. We hope that the justice, and both the UPLB and the UP community hope that the justice for the death of these modern heroes be served soon. So that's Sir Leonard, one of the foremost expert in botany, a brilliant taxonomist that pushed the Philippine taxonomy into its finest. The next issues that I'd like to mention, number 2, is that the environmental impact assessment. So environmental impact assessments or what we call EIAs. So the law and the law to our law, and of course it is the mandate of the DANR to enforce these environmental laws. So your DANR actually require companies, so say for example SM or Shell or any other companies that would like to build large malls, large Ferris wheels, large gasoline station, to provide us, to provide them, no, and to provide the people with what we call environmental impact assessment. So these EIAs are are assessments, or are somewhat paperworks or some requirements that these companies need to pass, no? These companies need to pass this assessment test. So EIAs are like assessments or tests, no, that these companies must pass before they are given permission to build their malls, to build their gasoline station, to build their drilling or whatever that is, to build the mines, no? And in that environmental impact assessment, I actually we actually try to scrutinize all of these environmental impact assessment. In that EIAs, there is a need for taxonomic information. So if you guys, all of you guys want to specialize in biosystematics, there is a real need for taxonomists in the DENR, and especially foresters in the DENR, to try to measure the level or the environmental impacts of different projects in our ecosystem. So yeah, that's the first lucrative job that you guys might want to go to after this course, or after finishing your biology course here in UP Baguio. My colleagues once told me that for them to, when they're doing environmental impact assessment, and if you are hired by DNR, you can at least get 60,000 peso monthly as a compensation for the job, no? But my colleague also told me that there are some practices, no, some weird practices, if I say so myself, that the DNR are, or the companies, private companies are doing, no? Yung tawag dito ay panunuhol, mga ganon. Ang gagawin, ang problema kasi sa mga EIA, bayad kasi ng yung company mismo, kuwari yung SM, magbabayad talaga sila ng pera. O magbabayad sila for the EIA. So hindi to gagawin for free ng DNR. So magre-request yung company na to for EIA. Babayad sila. So popondahan nila yung mga scientists, pumunta dun sa lugar para i-assess yung lugar, kung pwede nga bang putulin yung mga puno at lagyan ng mall. So yung mga nag-a-assess at gumagawa ng environmental impact assessment sa DNR ay parang naiipit dun sa sitwasyon na gusto nilang ayusin yung trabaho nila at i-assess talaga na maayos. But since they are funded by SM or the private company, so parang hindi din nila ma-publish na fully, no, ng maayos. So may mga ganung issues pa. But yeah, if you're good in taxonomy, you can join that force and try to provide that taxonomic information. What species are going to be affected, what species might be reduced to ashes by building these massive things in that forest, for example. So that's your EIA. The next issue that I'd like to mention here is the issue on of conservation of your um, species. So there's a need for a what we call precise baseline data, no? Precise baseline data for your species. Because uh, before we can conclude if a species is extinct, if a species is nearly threatened, if a species is endangered, so on and so forth, before we can manage these species out there, we need to have that baseline data. We need to have the data on their numbers. So we can't know their numbers, no, if we don't know them at all. 'Di ba? Yun yung pinaka-conundrum natin. So, paano mo aalagaan at i-conserve at i-manage yung mga bagay-bagay kapag hindi mo sila kilala? You can't protect what you don't know, ika nga nila. So, yun yung tatlong issues, no? Yung kakulangan ng tao, the lack of new people joining in the force, the need for taxonomic data in environmental impact assessment, and the need, no, the critical need for baseline data, species data, precise species data in conservation management. So, my solution for you today is molecular systematics. It's the only solution that is coming across my mind right now. So, why do molecular systematics? Well, if you perform molecular systematics instead of the normal systematic work where you go into the field, you fetch those plants, you identify the plants, and you try to describe each species that you collect, you describe their morphology and so on, you go to those nitty-gritty process. Instead of doing that, you can just do molecular systematics, where you can speed up the process of describing the species. And you can actually, in just one simple laboratory work, you can deduce up to the species level and the genus level the identity of your specimen just by doing some simple lab work. You don't have to go through all of those tedious process that we mentioned earlier, no? in identifying species. So yeah, one simple example of molecular systematics that I had experienced also is what we call the barcoding of life. So CIBO, or Consortium for Barcode of Life, proposed the use of what we call barcodes. So barcodes are these simple regions in your DNA. You know, these regions, no, these are short standard gene sequences in your DNA that you can use as a standard, kind of like a barcode, no, for your, if you remember the barcode in your grocery stores, it kind of works like that. So if you scan this barcode, it will mean species 1. If you scan another barcode, it will mean species 2. No, it kind of works like that. These are specific standardized gene sequences in your DNA, in the DNA of every organism, for example, of most organisms, that you can use to determine the species, to identify the species. Okay? So the particular barcode that they use, no, here is what we call the COI gene, or your dun ko na isulat, cytochrome. Mali na pagsulatan ko. Cytochrome C oxidase. Okay, so cytochrome C is, or cytochrome C oxidase 1 is a mitochondrial gene. So it's in the mitochondrial region, no? Mitochondrial gene region. So the CBOL, or Consortium for Barcode of Life, used that particular gene sequence to, as a barcode, no? As a reference region, to try to identify and to assign a specific barcode for every species out there. Why do that? Because, well, since most of your organism have mitochondria, if you compare all of those mitochondrial COI, or your cytochrome C oxidase region, gene region, you can deduce their identity. So that's how they do it, using your COI gene. So the one that I experienced, or I used back then in my RA days, my research assistant days, we were working with, not with COI gene, but with MATK gene. So scientists can choose different gene region. No, well, in that case back then, I was using different DNA purifying mechanism, different DNA kits, and I was trying to extract some DNA in this particular plant. And that was so difficult to extract the DNA from, and we were trying to create a barcode for that plant so that we can reference it in the other barcode in the database. So yeah, that's how it works. So you choose a barcode, you compare the barcode to the others that are out there in the database, and you can see how this will work perfectly, and how this will work well, because you don't need, if you do this, if you use the barcode for that organism, you don't have to describe the species anymore. You just need to extract their DNA, and voila, you can get the identity of your organism. That's how simple it is. That's how speedy the process is. It's kind of like a shortcut, actually. So why use DNA? Why use DNA in molecular systematics? Why use DNA in assigning identity into an organism? Well, first of all, there are much more molecular characters that are available for our analysis than morphological characters out there. The common example that I always explain to my student, that I always give to my student, is height. No, so imagine you have a tall person with a very big neck right here, or imagine you an average size person, or a midget, or a small human being. Okay, so imagine the character height, a very simple character that is your height for humans and for plants, for example. So height for us humans is actually coded by not just one, not just two, but a couple of genes. There are a couple of genes that is responsible for your height. These genes are scattered across your 20 plus chromosomes. They're scattered all around those chromosomes. Not a single gene is responsible for your height. These genes contribute a little by little to your height right now. So that's the first example. So if you're using just your molecular character that is height, you just have one. But if you go to the molecular characters, the molecular genes that are causing that height, you have multiples of genes that you can now study. So definitely there are more molecular characters out there than your morphological ones. And not to mention your DNA, not every part of your DNA is actually expressed. Most of your DNA, what we call the introns and the exons, diba? Kung maalala nyo, most of your DNA are kind of like fillers. But scientists are now thinking that these are not just fillers. These have some functions too. But yeah, there are a lot of sequences out there in your DNA. Okay? Now, the second reason why we use DNA is that it is easier to define a particular character than to use your morphological one. No? It's, say for example, it is much better to use letter differences, comparing letter differences between species 1, between you and your brother, between you and your sister or someone, between you or your ancestors, than to compare your morphology. Why it is much easier to define? Because it's not that ambiguous. Whereas when you refer to the morphology of that organism, you will need to set it is pink, it is white, it is pale, it is that person is brown. How can you say it's brown? It's or what degree of brownness is brown? It is brown level 1, level 2, or something like that. It is much more ambiguous when you use morphology compared to just using that letter differences in your DNA, diba? Now, the third one of why we use DNA is that the organism's evolutionary history is always documented in its genome. Okay? So, not all morphologies or not all phenotypes are passed from your parent to you. But your parent's DNA is almost always passed to you, diba? So, any changes along the way, along the progeny line, along your sons, your daughters, your granddaughters, you can track those changes. So, there are a couple of taxonomic issues that can be addressed by using DNA than just using your plain old morphological characters. The first one is you can resolve species boundaries, for example. Because when you use morphological characters, you can sometimes, not sometimes, and most of the times, morphological differences. Say, for example, in your plants, different plant species. When you're in the field, these I will tell you personally, you can't, that's it. You can't just compare different species that easily. Sometimes there are very subtle differences between different species. Sometimes different species would look very similar to each other, but they are of different species. They are of different genera or they are of different families, but they kind of look the same. Sometimes this is because of convergent evolution, wherein due to the same environmental condition, these species end up looking the same because they are subjected to the same condition, but they are of different ancestry, but they kind of look the same because they are exposed to that same environmental condition for that long period of time. So yeah, it is really hard to compare, even if you're a professional taxonomist, if you are a hard earned or a pro taxonomist, even if you are very old in the game of taxonomy, these subtle differences might pass your eye. No? You might miss these subtle differences in morphology if you're just looking in morphology. Sometimes there are differences in morphology of your organism, but that's just because they are of different life stages. No? This is one particular example is the eel. The juvenile stage of the eel is different from the adult stage. That's why some scientists are identified those different stages as different species, something like that. And sometimes there are this sexual dimorphism or sexual differences between the male and the female. So your male is kind of different from your female and something like that, but they are of the same species still. So that might fool you. But your DNA sequences doesn't lie. Your DNA sequences will provide that rich additional data for you to discern such differences between species. So these differences are not just, you can discern that you can conclude that these differences are not just caused by the difference in the life history stages of that organism. It's not just caused by the sexual dimorphism or the differences in the male and female of that species. You can even discern that very subtle differences in morphology just by using DNA. One example that I would like you to have here, and my experience in the field, is when identifying orchid species. When you're in the forest and in very dark, massive forests out there in Benguet or in Nueva Vizcaya or in Mountain Province and so on and so forth. All of those forests, no? In Pulag, yung mga bundok na inaakyat namin, pag nasa loob ka na nung forest canopy, hindi mo na makikita yung pagkakaiba-iba nila. Minsan parang bulag ka na kasi nasa tataas ng mga tuktok na mga puno pa, nakadikit kung saan-saan yung mga species na yan. Tapos, kailangan matalas yung mata mo. Ang kailangan pa nga dito, kapag sa orchid, lalo na sa orchid, kailangan may bulaklak. Kailangan may makita kang flowers. If you don't see any flower, you're just wasting your time. Because without the flowers, you will not be able to identify the organism. You can't work out the morphology of that organism. Because in most, not just in orchids, but also in most flowering plants, in most angiosperm, you need flowers. So yun yung technique namin. Kapag nakakita kayo ng flowers, if you have flowers, you can identify that organism. Well, if you work hard enough. You just need the flowers. But if you don't have the flowers, you're lost. You can't do shit without the, pardon my word, but yeah, you can't do shit if you don't have the flowers. That's the hard reality. But when you have DNA, you can just grab that organism, you can extract the DNA of that organism, and you have the identity of the organism without waiting for them to bloom. Okay? So the next one that we are going to talk here that your DNA can address, the next issue is your DNA sequences help assign species name to tissues. So there are many biological tissues out there that are collected, no? But since this is just some tissues, this is not the whole organism, you can't work out the identity of that, no? But with DNA, you can just pick some portion of that plant, pick some portion of that organism, and you can get the the DNA, and you can run it to the lab, and you can get its identity. It sounds so easy, but yeah, it's just like that. You don't need the whole plant, you just need a portion of the plant, you just need a portion of your blood, di ba? Laway lang nga yung kinukuha sa atin pag mag-ano tayo ng DNA test. So mga ganun, no? Another application for this is for the quarantine solution for conservation and food safety. Ito yung kwento ng canned meats, no? When you hear the stories of canned meats, not just in the Philippines but in US, you hear this conspiracy theory that the corned beef was made of human meat. Gawa pala sa tao, kinakain mong corned beef, Argentina, ganyan. No? Or CDO, nag-plug pa ako. But yeah, so kapag pwede mong kahit yung mga muscle fibers lang ng corned beef na yon or nung luncheon meat, no? Bye-bye spam, hello maling. No, pwede mo pa ring matest kung galing ba siya sa tao, galing ba siya sa pig, kung anong pig or kung galing ba siya sa beef or kung anong species ng beef siya galing by just extracting the DNA from that. No? So pwedeng ganun yung mga application niya. As long as hindi na-destroy yung DNA dun sa sample ng canned meats na yun. So yeah, it can be used for food safety too. So yung mga ganung issues can be addressed by using DNA. So you don't need the whole organism to identify it. You just need a certain tissues of that organism. You can run it to the lab and you can identify it. So there are different DNA-based systematic methods out there. So usually these are based on the type of genetic material that we're dealing with. So there are a lot of genetic material out there. The first one is what we call, so let's divide this into two. So we have your genomic and plasmid, the first one, genomic and plasmid DNA. And we have nuclear, mitochondrial, and chloroplastid DNA. Okay, so what's the difference between them? So mostly your genomic DNA and your plasmid DNA is applicable for what you call your bacteria, your prokaryotic organisms, your prokaryotic organisms. Your prokaryotic organisms have, if you imagine them, they have these two types of DNA, kumbaga. They have two types of DNA inside them. They have what we call the genomic or the chromosomal DNA inside them. So these are the larger bits of information inside them. So kapag ito yung bacteria mo, it has the chromosomal DNA. It doesn't have a nucleus, di ba? Kung maalala nyo, prokaryote yan. So it has your genomic DNA, something like that. So this is, of course, vital for the cell division. So this is a larger chunk of genetic material inside your bacteria. This is responsible for most cellular activity of your organism. So it codes for proteins, it codes for the cell wall, and so on and so forth. It has associated proteins that holds them together. Okay? So this is your chromosomal or your genomic DNA. Now, how does your genomic DNA get passed to the offspring? Of course, you need to replicate that DNA and you need to divide it evenly. Diba? You need to replicate or duplicate it and give it to your progeny. So you need to replicate it first and give it to your progeny afterwards, after division. But you need to replicate it. So it is transmitted through cell division, usually. Yes, through cell division. So, di ba? Maalala nyo, kailangan mo nang doblehin or i-duplicate yung genetic material na nasa genomic DNA mo. And then, after the replication period, pwede na mag-divide si cell at equally maibibigay, no, yung same genetic material dun sa kanyang mga offspring pag nag-binary fission na siya. Okay? Now, there is this other type of genetic material inside your organism. This is what we call the plasmid DNA. So this is a different kind of genetic material from your so this is your genomic DNA. This plasmid DNA looks something like this. These are circular bits of information. So I call them as such, no? Because they look like your plasmid DNA are circular bits of DNA. Of course, since they are smaller in size, they contain less information and they have no proteins that are holding them or clumping them together. But the key thing with your plasmid DNA in your bacteria is that this plasmid DNA is responsible. The information usually that is contained in your plasmid DNA is responsible for antibiotic resistance. So antibiotic resistance, antibiotic tolerance is a big problem right now in our society, of humanity. It's a big problem of humanity. The first antibiotic that we use was amoxicillin, di ba? Penicillin. Yung mga amoxicillin, mga ganyan, yung mga una nating ginamit. Amoxicillin, penicillium from the fungi Penicillium notatum. But now, hindi na gumagana si amoxicillin. Hindi na gumagana yung penicillin na yan. Mahina ng klaseng antibiotic yan. Yung mga mamahaling antibiotics na ginagamit nyo ngayon, yung amoxicillin with clavulanic acid, mga ganyan, yung streptomycin, at kung ano pang may mycin. So, malalakas na klaseng antibiotic na yung mga ginagamit natin. Hindi na gumagana yung penicillin ngayon, no? Diba, nung bata kayo, siguro, dinidikdik pa nung nanay nyo yan, tapos ilalagay sa sugat nyo yung penicillin ngayon. Wala nang kwenta yan ngayon. Kasi nagkaroon na ng antibiotic resistance yung mga bacteria natin. So, yun yung problema. And scientists found out that antibiotic resistance, the gene for antibiotic resistance, ay nandun sa mga plasmid DNA natin. At ang problema nito, guys, yung plasmid DNA compared to your genomic DNA. So again, kung ito si bacteria mo at ito yung anak niya, yung genomic DNA mo, vertical yung pag-transfer ng information. So, yung DNA nito, yung genomic DNA mo, kailangan i-replicate, tapos hahatiin dun sa kanyang dalawang anak. But, for your plasmid DNA, so vertical, okay? But, for your plasmid DNA, kahit magkapatid, no? Kahit magkaibang lahi, yung mga circular bits ng information na yan, kahit nasa environment niya, pwede siyang pumasok or ma-transfer dun sa organism. So, usually, horizontal yung gene transfer dito. So, hindi siya vertical yung transfer, horizontal yung gene transfer dito. So, hindi mo kailangan ng mag-divide at mag-replicate para mapasa yung antibiotic resistance. Kahit yung mga kapitbahay na cells, na bacteria, ng bacteria na merong antibiotic resistance, kaya niyang ipasa. Kaya huwag kayong magtatapon at huwag kayong mag-overdose ng antibiotics. No? Kasi yung entire gut bacteria nyo, madidevelop nila yung antibiotic tolerance, yung antibiotic resistance kapag basta-basta lang kayong umiinom ng antibiotic kasi may ubo kayo, may sipon kayo. Virus naman yun, hindi naman yun. Hindi yun bacteria, no? So, mga ganon. So yeah, yun yung kaibahan ng genomic DNA and your plasmid DNA. Ang dami ko ng kwento. But yeah. So next is, for most eukaryotic na organisms naman, we are dealing with what we call nuclear, chloroplast, and mitochondrial genomes. Of course, your chloroplasts are mostly found in your plants, but also found in algae, for example, yung mga lumot-lumot na yan, yung mga naaral natin sa readings, di ba? But yeah, so there are differences between them. And from your knowledge of your from your knowledge of our reading, why are they different from each other? Because the nuclear DNA is the DNA of that organ, the DNA of the organism, the host. But your mitochondrial and your chloroplast DNA is different. Why is it different? Because these organism, your chloroplast and your mitochondria, is a different organism. No? It's a different organism that was eaten back then through the process of endosymbiosis. So magkaiba yung DNA nila. It is even replicating independently of the nuclear genome inside. So yeah, kung pag kinumpare natin sila hand to hand, no? Maliit ang chloroplast at mitochondrial DNA mo compared dun sa nuclear DNA mo, no? Dun sa nuclear genome mo. And the origin of your chloroplast, diba? Kung maaalala natin, yung primary and secondary endosymbiosis natin, the origin of the chloroplast is the cyanobacteria or a version of a cyanobacteria that was eaten. Okay? Now, for your mitochondria naman, it is theorized that it is a purple bacteria or a version of a purple bacteria that was eaten during back then. No? So yeah, so cyanobacteria here, bacteria here, the origin, no, of your mitochondria, a purple bacteria. Now, your nuclear genome, of course, this varies from different species. If you're a human, you have a different nuclear genome from, say for example, an insect or a plant, no? Say you have a different genetic history. So different origin yan, of course. And usually napakalalaki ng mga nuclear genome na to. Okay? Okay. Now, in terms of inheritance, pangit naman ng E. Lagyan natin. E. In terms of inheritance, your chloroplast genome, your chloroplast DNA are inherited maternally. Of course, your nuclear DNA is biparentally inherited from your mother and your papa. Diba? Kalahati-kalahati si mother and si papa. Yeah. But kapag pinag-usapan naman natin si mitochondria, medyo ano siya, medyo weird yung inheritance niya. Well, sabi dito, no, maternal siya. But bakit nga ba maternal yung mitochondrial, well, kung sa tao, no, bakit maternal yung way ng inheritance or yung way ng pagpass ng mitochondrial DNA? Well, most of your mitochondrial DNA comes from your mother. Because remember, during fertilization, it is in the egg cell, no? Yung egg cell mismo ni mother mo, yung egg cell mismo, so yung egg cell mismo ni mother mo, so palakihin natin. So yung egg cell ni mother mo na haploid ay meron siyang mga maliliit na mitochondria inside it. So yung egg cell ay may mitochondria inside it. Okay? Okay. But, so, the mitochondria inside the egg cell exists, no, even before fertilization. But scientists have reason to believe that there are some few mitochondria also in the sperm cell. No? Because from your reproductive science, kung maalala nyo, your sperm cell needs some energy to propel itself and swim in the vaginal tract of your female. Kaya nga lang, kaya Kaya sinasabi din ng mga scientists mo na, so, kung yung sperm cell mo, ito yung sperm cell mo, yung mitochondria daw niya ay nasa dito sa bandang tail niya. Kasi kailangan niya panggawa ng energy, no, para makakapag-swim siya. But ang problema doon ay once na mag-meet yung sperm at yung egg, nagdi-dissolve yung tail part kasi ni sperm at naiiwan siya. Kaya most scientists believe that your mitochondrial DNA is transmitted maternally. So that your mitochondrial DNA comes from your mother. No? Because during fertilization daw, yung sperm ay nagdi-dissolve. Yung sperm, yung tail ng sperm at yung mitochondria na nandun sa tail ay nagdi-dissolve once mag-meet sila. So, ganun yung current theory nila doon. Kaya naglagay ako dyan ng question mark maternally. So, in terms of shape, circular DNA, and your nuclear genome is linear in forms. Remember from our previous discussion, the concept of molecular clocks. So, these molecular clocks uses a constant rate of evolution in some genes to estimate the time at which a certain evolutionary changes occur in the DNA. So remember from our previous discussion, if you have a gene right here, for example, so gene A, if gene A mutates at a certain constant rate of 1 base pair per 1,000 years, so it mutates at this constant rate, no? It has a constant rate of mutation, 1 base pair mutation, since it's a somehow conservative gene. It only mutates about 1 base pair per 1,000 years, and you see a fossil record. You see an organism. A fossilized organism, and you compare it. So this is a fossilized, let's say, human or fossilized Homo or hominin group. Or a much clearer example would be, I guess, your Neanderthal remains, no? A hominin Neanderthal remains. And you compare it to the modern anatomical human, say for example us, and you see that the modern anatomical human has more mutation on that gene A, and this fossil record has lower mutation on that gene A, you can conclude that the modern anatomical human is older because it accumulates this mutation. No? So the thing here that I want you to look is that we are trying to find that molecular clock, that molecular marker that we can use to extend our phylogeny, or to extend our understanding of this evolutionary change, not just to what we have now, not just to our ancestors, but to the fossilized records out there that we can see. So we need to make an assumption about how often this mutation rate occurs over time. Now, to give you an idea of how difficult it is to find that sweet spot, that molecular marker to use to track that evolutionary changes over time, I'd like you to think about this, okay? That genes mutate at a different rate. Now, say for example, a gene that codes for a certain vital enzyme. or a structure in your cell, no? If that gene is super important for you to survive, usually that gene is conserved. If that gene is super important for you to survive as an organism, usually there will be zero or a very low, no? Very low chances of mutation. Because if that gene mutates, you're dead. That's it. If that gene mutates, if you fuck with that gene, you're dead. Okay? You're gonna die if that gene is ruined by this mutation. So usually these vital genes, no, that we are referring to are conserved most of the time. Now, when do we use genes with slower rate of mutation? We usually use these genes with slower rate of mutation for older groups. Because if we use faster genes with faster rate of mutation, we can't see the changes, or we can't extend our conclusion, and we can't see the changes for those organisms that are way back, no, those organisms that are way older. Now we use the genes with a faster rate of mutation to assess the relationship naman between a closely related population. Why do we do that? Because with the genes with a faster rate of mutation, you can see tiny differences, no? Even in your closely related population. Okay? So, in deciding what genetic marker or what gene we are going to use, we need to look at the rate of that gene, the rate of the mutation of that gene. Okay? So we can use the appropriate gene to assess the evolutionary relationship of this organism. So we have what we call the neutral theory of molecular evolution. So neutral theory, basahin natin dito, sabi niya, neutral theory states that much evolutionary change in genes and protein has no effect on fitness and is not influenced by natural selection. So anong ibig sabihin niya dyan? Ang ibig sabihin lang niya dyan that most variation, no, at the molecular level does not affect fitness. And therefore, the evolutionary fate of the genetic variation is best explained by what we call stochastic or random processes rather than what we call the natural selection. So the molecular variation the variation at the molecular level is best explained by stochastic processes rather than your natural selection. So yun lang naman yung sinasabi niya dyan. Okay? So yeah, at the molecular level, your variation is driven, no? The evolutionary change at the molecular level is driven by random stochastic processes and by genetic drift rather than the natural selection. Okay? So, ang ina-assume natin with neutral theory is that the mutation at the molecular level are neutral. Okay? The mutation at the molecular level are neutral mutations. So pag sinabi nating neutral mutation, or pwede ding tawag natin dyan is silent mutations. Ibig natin sabihin yan, these are the mutations that does not affect the organism's ability to survive and reproduce. Okay? So, the neutral theory assumes that most mutation that occurs are neutral in nature rather than deleterious. Siyempre, pag sinabi mo namang deleterious mutation, ito yung ikamamatay talaga ng organism. So, bakit natin ina-assume na neutral mutations yung nakikita natin? Kasi kung iisipin nyo, kung deleterious mutation yung nag-occur, wala kang makikita ang organism, di ba? Kasi nga, namamatay yung mga organism mo na yan. So, therefore, ang makikita mo lang ng mga mutation sa mga nag-e-exist na organism na sini-sequence mo ay yung mga neutral na mutations. Of course, pwede ka rin magkaroon ng beneficial na mga mutations. But here, ang ina-assume natin with the neutral theory is that most mutation are neutral or has no effect on the organism's ability to survive and reproduce. Okay? So that's your neutral theory. Now moving on. There are problems with how gene mutates or the rate at which gene mutates. What kind of problems are we talking about here? We're talking about how slow or how quick the genes are mutating. What problems do arise from that? If, say, for example, your genes are mutating very slowly, ibig sabihin yan, wala kang makitang variation. No? Kasi nga, sobrang bagal ng mutation, halos wala ka ng variation na makita between species 1 and species 2. And because of that, dahil napakaliit ng variation na nakikita mo between species, lumalapit na tuloy siya dun sa sequencing error. Dun sa error, itong sequencing error rate natin, ito yung error ng machine na ginagamit natin or ng system na ginagamit natin. So parang dahil nga wala ka nang makitang variation, napakaliit ng mga variation na nakikita mo, lumalapit na siya dun sa error ng system at ng machines na ginagamit natin pangkita ng mga changes na yon. So parang wala na din kwenta siya. Parang unreliable nang gamitin siya. Kaya napakabagal no rate ng mutation. Halos wala ka na makita ang differences between them. So mas malaki pa yung error, yung variation na ikita mo dahil sa sequencing error, dun sa mga error na nire-register ng machines mo. No? So yun. So pangit na gamitin yung sobrang bagal na mag-mutate ng mga genes. Ngayon naman, kapag sobrang bilis naman, no? Kabaliktaran naman ng kanina, sobrang bilis na mag-mutate ng genes natin. Ang problema naman yan, nagkakaroon ka ng tinatawag nating parallelism at yung mga tinatawag nating reversals. So, dahil nga sobrang bilis mag-accumulate ng mutation, ang bilis din magbago ng mga nung tinitignan mong characters. So, at the expense na mawala na yung tinitignan, parang nabe-blur out na yung tinitignan mong phylogenetic relationship between them. Okay? So, yun naman yung problema kapag sobrang bilis ng rate of mutation.